pictures alone, though, in culture, you can't prove what virus you found yet.
Of course, electron microscopy is not sufficient to prove you have a retrovirus. That's clear. You need other characteristics like the density. The reverse transcriptase activity is the key enzyme, which is at that time was only associated with retroviruses. That's how we describe it first. We didn't describe the electron microscopy as a proof. We described the reverse transcriptase activity as a proof it was a retrovirus. But here also we were criticized by some people from Gallo's lab saying, are you sure you have a reverse transcriptase activity there? Could it be a cellular DNA polymerase? We had this kind of question.
Could we take their blood, get their T cells, like we would do with like the leukemias, like we did with the leukemias, the same protocols we did with leukemia. Take those T cells, clean them up from other cells, grow them with interleukin-2, our old friend, and see if we could find any evidence of release of virus particles, and if we were right that they would be retroviruses. And how to do that was with a surrogate marker. You don't use electron microscopy in those days, except maybe 1 or 2 pictures just to confirm or to see the structure of this particular retrovirus. The assay was reverse transcriptase.
And ask Baltimore or Vermouth, and I'm sure they will confirm that reverse transcriptase activity is a characteristic of retroviruses, but it's not specific to them.
What do you mean when you say reverse transcription is a characteristic of retroviruses, but not specific?
Oh, let me give you an example. Hair is a characteristic of humans. Black, white, or yellow, we all have hair. But it's not specific because there are many animals, for example, cats, dogs, which also have hair. And finding a hair in a room, it doesn't mean that a human being was there, or a cat or a dog.
Are retroviruses the only ones that can reverse transcribe?
No. There are other forms of reverse transcription that are used in various ways inside the cell. For instance, the ends of chromosomes are made by a reverse transcription process. That's how they're maintained stable. There is reverse transcription in the inheritance of all of our cells, which comes about from endogenous genetic elements in the cells or in the cells of our ancestors. Because once that information gets into our cells, into our genomes, it stays there forever. So it could be that we've inherited information from monkeys or from other animals that are in our lineage. And so, no, reverse transcription is actually very widespread. Something like 50% of the DNA in our cells comes about by reverse transcription.
That much?
Yes.
Oh, wow. Okay. I thought it was 1 to 2%. Going back to, I thought it was 1.5 to 2%. The 50%, the retroviral DNA, or retroviral genes in our DNA, what biological function do they serve?
Now, we got to make a distinction. When I said 50%, I'm saying 50% of the DNA came about by reverse transcription. But it's not all retroviruses. Lots of it is just repeated elements that are there as what we generally consider to be parasitic DNA. DNA which is, as other people call it, selfish DNA. DNA which is in there because it's able to copy itself and reintegrate itself in other places. And this is something that's going on all